Hello, everyone, and welcome to another Terra training module. I'm Frank Ambrosio, a bioinformatics scientist with Theogen Genomics, and in this video, we'll be going over the process of running Mercury batch on Terra.bio. Before running the workflow, one must have uploaded some metadata using the metadata formatter. If you have not completed this step yet, please check out our protocol at protocols.io about SRA and GenBank biosample link submission, or reach out to our support email, which is support@terrapublichealth.zendesk.com, and we will include a link to both of those in the description of this video. Now that we've confirmed we have our metadata uploaded to Terra.bio, let's jump right in and run Mercury prep. So the first thing we need to do is navigate to the workflows page in our Terra.bio workspace, and then identify and click on the Mercury prep workflow that we would like to run. In this case, we're running Mercury paired end prep because we have Illumina paired end FASTQ files. We do offer a Mercury single end prep. So if you need to import that workflow, please check out our video on importing workflows to your Terra.bio workspace. So let's go to the Mercury paired end prep workflow. And here we see some options. The first option is selecting the version of the workflow. We want to select the latest version unless we have a specific reason for selecting an earlier version. The next option is we select our root entity. In this case, we're going to be working with our Illumina paired end data. So we're going to use our Illumina specimen data table in Terra. Next, we must select the samples that we would like to run. Here we can filter samples by collection date or by sequencing run. In this case, we're going to be selecting all 10 samples. So we can either use this checkbox or we can use the drop down, select all, and click OK. Finally, we need to assign our inputs. So here we can use the familiar this dot notation in order to identify inputs. So here we'll use this dot assembly FASTA for our input. Once again, these samples must have been run through one of the Titan genomic characterization workflows, in addition to having the metadata uploaded. So if you have not run these through the Titan genomic characterization workflows, you will not have an assembly FASTA available. So you must have run them through Titan Illumina paired end in this case. So we have that assembly FASTA available to us because we see it in the dropdown. This means we have successfully run a Titan genomic characterization workflow previously, so we are good to go. Here we need to indicate our mean coverage. So this will be assembly mean coverage, our assembly method. And this is all going to be a little bit different if you decide to use your Clear Labs assemblies. So when using a Clear Labs instrument, you're provided both the assemblies and a FASTA file by Clear Labs. And many labs have found that it is beneficial to them to use the Clear Labs FASTA because the Clear Labs assembly process has some proprietary error correction regarding the boundaries of deletions. So in that case, you will be able to use the Clear Labs FASTA in this first input. So as we see here, I am just entering in all of the required fields. So to speak on the using the uh, Clear Labs FASTAs a little bit more. So the Clear Labs FASTAs, they do have that error correction built in. However, the entire pipeline is proprietary, meaning as opposed to the Titan workflows where you can go on GitHub, you can see the exact code, you can see all of the internal tools that were used, whether they be a mapping tool, an alignment tool. You can see all of those, and you can see all of their references and the literature, both explaining and detailing these tools and citing these tools used in future analyses. So it's really up to uh, the submitting lab whether or not they would like to use an open source tool that does not have that proprietary error correction built in for the boundaries of the deletions, or if they would like to use a fully open source tool where they can look up the code, they can share the code freely, and they know everything going on underneath the hood. So here in the Mercury Prep input section, you will only need to do this once, hopefully. Once you do this, you will be able to save these inputs and they will persist in your inputs page until you need to change them. This is part of the reason that we include both a single end and a paired end version of this workflow. In the event that you would like to have one of these saved with all of your inputs for Illumina and one of them saved with all of your inputs for either Oxford Nanopore data or Clear Labs data. So here I'm selecting the dehosted inputs. These dehosted reads are actually the reads that we're going to be submitting. And by dehosted, we mean that they have had their host reads removed. So any of the reads that map to the human genome have been removed using the NCBI human scrubber. Um, if you do not have dehosted reads available for whatever reason, there is an option to have NCBI scrub your reads for you. If you have any questions about how to do that, please reach out to our support email, which is support@terrapublichealth.zendesk.com. Uh, all right, we are just about done with our required inputs, submitting lab address. Other optional inputs that we want to include. Let's see, purpose of sampling. Purpose of sampling and purpose of sequencing. Oops. And remember, these are the two fields that are being used to tag samples for performing some deeper statistical analysis later on. Let's see, do we have any other? Yes, we have our amplicon scheme, amplicon primer scheme, and the primer size. All right, so once you have all of these inputs saved, go ahead, click save. Once you have them all saved, you go to the outputs tab and you select use the defaults. You can click save again. And now, the next time you load this workflow, all of these inputs will be saved in these fields, so you won't need to resubmit them every single time. Almost forgot the submitter email. Definitely want to include that. Okay, gonna hit save again. Our outputs are saved, our samples are selected. We can go ahead and run the analysis. So let's say this is our Illumina paired end training data. And we are running this on Mercury Prep. Okay, you go ahead and launch this analysis. You should be able to monitor the status in the status column. Otherwise, go to the job history page to monitor it further. If you would like to take a deeper dive into monitoring the status of a run, please see our video on that topic. Otherwise, if you have any questions, concerns, comments, please reach out to support@terrapublichealth.zendesk.com. Or if you'd like to follow along with the protocol, we will include a link to the protocols.io page in the description of this video. Thank you all so much for watching. We really appreciate it, and we hope to see you again soon. Thanks.